Hey everybody, Mr. Murphy here. I'm going to walk you through chapter 15. I'm going to do this a little differently than previous chapters. I'm going to cover the whole thing in one, hopefully not too long video, but I'm going to go through like a lot of slides very quickly just to highlight some key terminology and talk about some of the more important aspects of it. So this is all about sort of the introduction of the concept of evolution. We understand that things change over time, and that's really what evolving fundamentally means. The idea of evolutionary theory is seeking to understand the mechanisms of how that actually happens. There's a lot of potential use, not just in terms of understanding sort of where things came from and how we have the diversity that we have in the world today, but also in terms of like artificial selection in the development of crops and new foods, industrial processes, understanding the diversification of life and how species interact. There's a lot of ecological gain that can happen from understanding the evolutionary principles behind sort of the arrival of certain traits and things like that, how organisms interact with their environment. So it's an important topic for a whole lot of reasons. This is a good slide just to remind ourselves that the word theory doesn't mean the same thing in our everyday sort of colloquial usage versus scientifically. Evolutionary theory is a well-tested and demonstrated sort of understanding of how we are able to observe the things that we observe today. It is a long history to the idea of evolution. So just, don't fall into the trap of, oh, it's just a theory or things like that. But we're not like that. Even before Darwin, there were lots of people who had ideas and written about this. We'll talk about one guy, Alfred Wallace, who is kind of he's the co-owner of the whole idea of evolution and natural selection, although in like high school we don't talk about him for some reason, but so lots of people involved in this. So Charles Darwin being sort of the big name that we talk about, he went on a whole long journey on the HMS Beagle, which we've seen this map and how this gave him a unique perspective compared to a lot of other scientific individuals, just because he was able to actually firsthand observe organisms in a whole variety of different ecological settings, and it really helped him to develop a better model for explaining why organisms are different and why organisms seem to all fit their environment. If you look at almost any organism today, you can run down the list of different characteristics that it has, and you can really pinpoint like what does that do for it. We don't see too many examples of organisms that are have some aspect to their biology or their morphology or whatever that doesn't make sense in the context of evolution. So this led to Darwin developing what is known as the theory of natural selection, which is the mechanism that would lead to this evolutionary change over time. And then, like I mentioned, Alfred Wallace actually had basically the identical idea, and they corresponded with each other around the time that Darwin published The Origin of Species. So it's important to remind ourselves that in the context of this, The Origin of Species was 1859. Mendel, meanwhile, was doing his work with pea plants and sort of figuring out the basis of inheritance. There's an interesting story about how Mendel's work kind of was lost to history for a time, and how it wasn't until later that in the 1900s that was sort of rediscovered and then contextualized with the established theories of evolution. And then, as you know, that would later be further supported by Watson and Crick, Rosalind Franklin, and the whole crew of people we've talked about in the development of the understanding of the genetic basis of passing on information between generations. So when we talk about evolution, an important thing here in italics, individuals don't evolve, populations do. So we have to be careful and check ourselves when we talk about evolution, because oftentimes just in like the casualness of our language, we say things like, oh, this animal evolved XYZ. And when we say that, hopefully in the back of our heads, we know we're talking about populations and we're talking about, extended periods of time. But, it's worth just sort of highlighting that, that's an important thing. Mutation is the main driving factor. And mutation, as we know, is changes to an organism's genetic code. And those changes can take a lot of different forms, and those changes can be positive or negative or neutral. But that's the basis by which change originates. And so that's why, again, we're not looking at this on an individual basis, because once an individual is born, it is set. It has its genes, and we could get into epigenetics and how life events can sort of modify things. But ultimately, it has the code that it's going to have, and so it's not going to change. It's not going to change, the DNA on chromosome 12 so that it has some new ability or anything like that. That's not how evolution works. So mutations occur between generations, and that builds up over time. And we've talked in class about how one of the hardest concepts, I think, or parts of evolution for people to really wrap their heads around is just how much time we're talking about. We live in a human mindset of what is our perception of time. It's hard for us to really grasp what tens of millions of years or beyond can really mean in terms of the passage of time, since all of human history is 20,000 years or 30,000 years, at least in terms of interesting stuff. Those mutations, as I said, could be deleterious, which is where they are harmful to the organism. They can be beneficial or have no effect. And because of that mutation, we get different forms of a gene existing within a population. And this is key. So since oftentimes these are neutral changes on their own, having a slightly different phenotype for a physical characteristic isn't necessarily good or bad until you apply it to an environment. And so that's where this idea of the gene pool comes in, which is looking at a population and saying, okay, well, what are all the different alleles that exist in this population? And the frequency of those alleles then is how predominant or how much of one particular allele out of the total allele population do we see. So here's a typical just representation of a gene pool. So a lot of Darwin's work came from looking at the natural world and his journeys, but then also through artificial selection of plants, in a very Mendelian fashion. He performed experiments involving selection on a variety of different plants, and one notable example is this wild mustard plant, which over time had been differentiated into all of these different things that we've probably eaten and never really considered the fact that they came from a common origin, and it was only through intentional selection of particular traits that they developed into a whole variety of foods that we eat. If you're interested in a little bit more reading on this subject, looking up about Brussels sprouts is kind of interesting because probably not within your lifetime, within my lifetime, Brussels sprouts have undergone pretty significant change, such that, when I was a tiny kid, Brussels sprouts were pretty fundamentally different than they are today. And that's why if you talk to your parents or grandparents, they may sort of remember hating Brussels sprouts, but then maybe they love them today. And a lot of times people attribute it to it's because I fry them up and they're real tasty that way. But actually the actual plant itself has been undergoing significant selection, artificial selection, to make them much tastier today. It's kind of interesting. Darwin also bred pigeons because why not? And I'm pretty sure Darwin took all these pictures. These look like very Darwinian pictures of pigeons. But again, just as an example, dogs are a great example of this as well. The existence of all the different breeds of dog in the world, that's completely artificial selection. So that's intentional choice of particular traits in deciding which specific individual is going to be involved in breeding and leading to subsequent generation. And that leads to the ability to hone in on particular traits and really exacerbate sort of their significance. perhaps to the detriment of the animal. I don't want to get into like a controversial thing, but there's a lot of species, breeds of dog that have pretty significant problems because of this heavy artificial selection that happens. And if you're somebody who's interested in like breeding dogs at some point, there's a lot more to it than just like, let's get some dogs and put them in a fenced in area together. There needs to be a whole lot of thought and care put into that in order to ensure that you're making not just the purebred whatever that you want, but healthy offspring as well. Laboratory experiments have demonstrated this as well. Fruit flies, again, great model organism. Can be seen that with selection for more or fewer of these abdominal bristles, that pretty quickly over not too many generations, you can lead to a pretty notable difference in the predominance of those bristles. So if you have a vial of fruit flies and you pull out all the ones with tons of bristles, and then you put those in one container, and you take all the ones with the fewest and you put those in another, and you do that over and over and over again, you're going to change that population in a not surprising way. Which again, that's one of the frustrating things about evolution, and when people don't fully appreciate that, it's all really simple. It's just taking that idea and applying it now to nature. And sometimes nature separates populations, and nature allows populations to become notably different, and not quite as dramatic of a way as like physically pulling a fly out of one container and putting it into another, but it's not all that different either. And we'll get into that with later chapters. So natural selection, I'm sort of getting ahead of myself, is basically the fact that there's always going to be more individuals born in a population than can survive. And that's kind of the sad, horrible part of nature. But because of the differences that exist between these organisms, these individuals within a population, it establishes a basis by which a selection can naturally occur. That if you have a certain number of individuals, but you have a smaller number that can actually survive, get enough food, and have ample space and opportunity to survive long enough to reproduce, then what that means is that there's going to be competition, and that competition is going to be determined based upon which organism has sort of the right stuff to survive. And it's not to say that an albino squirrel couldn't survive long enough to reproduce in Pennsylvania. Sure, it could. By chance, maybe it doesn't run into predators. It doesn't have a problem despite having sort of a negative adaptation, or a trait that we would point to as probably being unhelpful. But in general, the squirrels that stand out more are in general going to not survive as long. And so that's where, again, you have to look at it on a population level, because individually, sure, some survive, but generation after generation, you're going to see fewer and fewer of those harmful traits. So these are adaptations, which are the new traits that form through mutation. And so that's kind of what I just said. Migration is another factor in this, that the flow of genes between different populations is a major factor. When we say migration, it doesn't have to be like geese, flying hundreds or thousands of miles or however far geese fly. It can be more subtle than that. It could be, a population of chipmunks, and they live on one side of a river, but sometimes, when there's a drought or something, some individuals can cross that riverbed and now be on the other side of the woods, and sort of be bringing whatever traits they happen to have and combining those with the population that lives on the other side. So there can be separations. Separations can exist down to the level of like one tree versus another tree, honestly. And so there's a lot of examples and different sort of scales to what migration and separation of different populations can actually be. Genetic drift is an important term to understand, and that's just the natural sort of random change that occurs. And so that's just on the basis that sometimes organisms are born with slightly different, mutations in their genes, and over time, maybe they're neutral, maybe they're whatever, just randomness in terms of survival. Like I said, with the albino squirrel who just by chance survives. Things like that just chance events that can't really be accounted for in terms of selection and things. In smaller populations, it's important to understand smaller populations are going to experience an enhanced version of genetic drift, and that's largely because without strong selection because of competition, you're going to see more of an influence of these sort of just randomly acquired characteristic differences between organisms. That can also be a huge negative thing in terms of the loss of functional or good alleles and characteristics, and it's part of the reason why conservation for different animals in areas impacted by humans, which is like the entire planet, why that's a important factor. An example is in India, populations of tigers, for instance, right? Tigers are a pretty violent and terrible organism in the wrong context. Like, you wouldn't want to run into one in your backyard. And so there are preserves where these tigers can exist, but because of the very confined nature of this, it creates very small populations, which then causes enhanced genetic drift. And so there's efforts there to encourage and basically create natural pathways for these tigers to exchange between these different preserves in order to allow for genetic drift and migration and things like that to basically level out. A population bottleneck is any event that causes a significant decline in a population in a relatively short period of time. And here's sort of the classic view of this. You have a bottle of jelly beans or something here, and if we say, okay, here's the population, and something happened, a volcano erupted, there's an earthquake, humans came in and made a development, something has vastly reduced the number of organisms that can survive. So now we have this smaller container of surviving jelly beans. And when those survivors then reestablish their population either at the same place or someplace else, what you're going to see is a pretty significant difference in the characteristics just by chance. These are bottleneck events are indiscriminate of alleles and characteristics. So these are typically natural disasters and things like that, where it basically wipes the slate clean, and then the few surviving individuals repopulate, and you notice that there's a pretty significant genotypic shift in that population. The founder effect is kind of similar, but it's just sort of a different basis. The founder effect is, imagine that you have one forest that is populated by whatever, and then you have maybe there was a volcanic eruption or something has created basically a new space that's newly fertile. It's newly able to sustain populations of organisms, and so you get butterflies from over here that move over there, and just by basis of the butterflies that happen to find their way to this new area and establish new populations there, that new population is probably going to look different in terms of gene pool allele frequencies than the original one, just due to the chance of which individuals happen to move to that new area. Some other terminology comes up, the idea of non-random mating. So we have, when we look at different populations, we have mating on a variety of different qualifications or whatever, depending upon the organism. Sexual selection, we have mates that are chosen on the basis of some physical characteristic or some interpretation of their health or whatever. And so you can see there's a trade-off between attracting mates and attracting predators. So you think like peacocks and different birds that have these like fancy feathers and stuff like that. Like, that's all well and good for wooing another bird, but then you've also got to deal with the fact that you've got all these crazy feathers and things, which maybe slows you down and makes you easier to pick off by predators. So there's a balancing act happening there in terms of those traits. Nothing's ever as simple as like, this trait's good, and so we just get more of that trait. There's always a trade-off in some way on some level when you think about these different adaptations, because at the end of the day, any physical trait that an organism has is a cost. It's a burden on that organism to develop the cells, to develop the chemicals necessary to like cause that trait. And so that's all well and good up until the point that it actually hinders the survival of the organism. So the bird example happens to be a pretty like in your face obvious example of giant feathers going to slow you down. We can see this in experimental studies as well. We saw this on one of our worksheets, this bird with the super long tail feathers, and basically that artificially lengthening it led to more success in the males fathering new offspring. So we can identify these different characteristics, and we can sort of exploit them through experimentation to understand more the basis of how strong of a factor is it in the success of the organism to have more young. Allele frequency is just the number of copies of allele in the population divided by the total alleles in the population. And there's some math in the textbook that we don't need to worry about. We look at the Hardy Weinberg stuff, which is coming up, but just to be aware that there's more mathematical tools to understand the distribution of alleles in populations and how we compare them and things. So Hardy Weinberg is basically the basis by which, if there was no influencing factors, we would expect the frequency of an allele in a population to remain the same generation after generation after generation. That means that this is an allele that in no way is influencing survival. It's not being selected for. It's not being manipulated by things like migration, immigration, mutation at a significant rate. This is just pretty like it's there, and it's just that's it. There's no basis of selection. We've talked about in class how that's not realistic to do on a population level or on an organism level, but on a gene level it is. We did the taste bud thing, the little taste paper thing. That doesn't have a basis on human survival. So the differences that exist in people as to whether you have genes that allow you to detect whatever sodium benzoate or whatever the chemical was, that's not going to influence your survival. And so that gene most likely exists within Hardy Weinberg equilibrium within people. It's there in some people, it's not there in others, and generation by generation, all things being equal, it's probably going to remain the same in humans. When we look at Hardy Weinberg, we consider this p squared, 2pq, and q squared representing the different genotypes. And we've gone through problems on how to solve these. If you need additional help with this, just Google Hardy Weinberg, practice problems or whatever. You'll find millions of worksheets that you could practice with. This is sort of showing the basis of the mathematics, which you're welcome to consider, but you don't have to. And why we come up with those equations for that. Like I said, populations never actually meet the conditions of it because all organisms are evolving. However, it's good for like individual genotypes or individual alleles looking at things that are stable. It also provides the basis for understanding when we're looking for evidence that something is evolving, that we can look at and establish that we see the allele frequency changing changing in the population between generations. So for instance, the fruit fly thing from earlier, the number of bristles or whatever. So when conducting that experiment, they're looking for basically what is the abundance of the different alleles and are they changing with each generation. Selection can happen in a variety of ways, stabilizing, directional, and disruptive. These are all pretty easy to understand. Stabilizing selection is where you have some trait and we're measuring that against fitness. Fitness is survival to the point of being able to reproduce. And you notice that the individuals in the middle here tend to survive the longest. And so if this is true of a population, then over time what we expect to see is that we're going to get a honing in on that trait, that whatever that middle level thing happens to be. This could be the size of the claw on a crab, or it could be the amount of pigment in the coat of a mouse. But whatever the case is, whatever the middle ground version of that phenotype is, happens to be the best. And so we see the population move towards that. It's easier to understand this in comparison to the other two types, because with directional selection, rather than seeing that bell curve sort of shrink in on itself, we see it actually shifting. And so in this case, if this was the claw size of a crab, maybe having a big claw makes them slower, and maybe there's some new predator that is good at catching these crabs or whatever, but the crabs can't, pinch them or whatever. And so we see that the next generation, next and next and next, we start to see a decline in the size of that claw, because maybe it makes the crabs more agile or easier to hide or whatever. I'm just making stuff up. But the basis of what directional selection is that change will happen, but that change will be directional, that you'll see a gradual shift away from whatever was sort of the norm is now shrinking or getting larger, or more pigment or less pigment, depends on the characteristic we're looking at. Disruptive is where you have this sort of middle ground, but now being in the middle is not good. And so being over here that's better, or being over here, that's better. And so we see that there's more fitness in the organisms that have the lower expression of that phenotype, or the greater expression of that phenotype. And so over time, you're going to start to see these populations diverge. And this is where speciation often kicks in, because when a population experiences a disruptive selection where it's either better to be over here or better to be over here, that really drives change in that population and may set the basis for those two sort of sides of that outcome to become increasingly different over time, to the point that they can no longer mate. Now, that takes a lot of time, and often takes a lot of really specific physical what am I thinking of? Like geographic influences, in terms of the ability of those populations to actually be separated, and it can continue to experience those selective pressures. So like the Galapagos Islands are a really good example of that, where you have these birds on different islands that are sort of honing in on different feeding styles, and then you just sprinkle in some time, and you end up with these finches that are notably different in key ways and can no longer reproduce with each other. Stabilizing selection reduces variation. So if disruptive is sort of creating this variation in the population, that you have one side and you have the other side, stabilizing is kind of the opposite. It brings them together, and it creates more sameness, which can actually be problematic if it happens to too great of an extent, right? Because diversity is what enables populations to overcome challenges. And if you have too much stabilizing selection of too many traits, then you get a lot of sameness between your individuals, which may make the population more collectively susceptible to change or disease or whatever. Human birth weight's an example of stabilizing selection. And so while this is kind of a sad graph to consider, mortality, infant mortality increases on the very low birth weight. It's kind of obvious, but also on the very large birth weight, just in terms of statistics. And so we see that humans have sort of honed in on a particular birth weight that tends to be the predominant outcome of pregnancies. Directional selection, it can create more extremes. An example is longhorn cattle, where we see, directional, where we see longer horns somehow leading to a benefit. I'm not like a super cow person, so I don't know if it's sexual selection. I don't know if it's the ability to defend and attack other cows are weird, because all the cows in the world are domesticated animals, so this is all like human selection anyway. So this may actually be an example of just pure artificial selection. You can check your book; they'll have more details on why they included this picture. But regardless, we see a selection for longer and longer horns, right? Horns, not antlers. I don't know, cows. Disruptive, I already kind of went over this, but the finch thing here, where you get the width of the lower bill a larger width, maybe that's better for cracking open larger seeds, and the smaller one's better for reaching into crevices and finding small seeds, or maybe insects or whatever. The mutations, quick change on section here. The mutations that can lead to these things we've talked about, but here's some other terminology. So nucleotide substitutions are the substitution of a single nucleotide, and those can be synonymous or non-synonymous, and these pictures help you to understand. Synonymous, that's like our silent mutations, where it doesn't change the amino acid it codes for, whereas non-synonymous can be any number of things. It may lead to a different amino acid, or it may lead to a complete stop of translation if you get a stop codon that appears in the middle of your sequence. And so the different types of genes that are experiencing these substitutions are show differences. So pseudogenes, which are genes that are no longer considered functional, tend to have more substitutions than genes that are relevant to survival, right? Hopefully that makes sense, that a gene, let's say, that is coding for some region that's no longer expressed or no longer relevant to the survival of that organism, it's going to be able to accumulate more differences over time because those differences are less likely to lead to devastating negative consequences for that organism. And so here you can see in the book, we'll have more details on the specifics of these values, but this is showing the rates of substitution for these different types of mutations, that substitutions occur in pseudogenes, which don't code for anything, versus synonymous substitutions versus non-synonymous. And the key here is that the non-synonymous, that's your changes in the amino acid, that's your changes to a stop codon. We see fewer of those substitutions per 10 million years than we do the other types of mutations, and for sort of the obvious reasons of the reason we don't see them isn't because they're not happening, but because they're often not survivable, and so they don't get passed on to the next generation. There's other types of mutations: insertions, deletions, rearrangements. The names kind of imply what they mean. These can have larger effects. They can change fundamentally the reading frame. That's the frame shift mutations. And then there's what's called neutral theory, which is that at the molecular level, the majority of variations are going to be neutral. Those neutral mutations are accumulated through just random genetic drift. It's just randomness because there's no selection happening for a neutral mutation. If the mutation is truly neutral, it's not aiding the organism or hurting it. So that's like a protein that had a slight change to one amino acid, but it still does its job just as well as it always did. That would be a neutral mutation. And if you were tracking sort of the accumulation of those neutral mutations, you would be looking at how that was influenced by randomness rather than any sort of selection. There's some math to support this, which I'm going to skip over, and just talk about sort of the end result is that we're looking at the fitness of these things and whether they increase fitness or decrease fitness. But all of this information about the different rates of substitution and the mathematics behind it and things is all about understanding sort of what's the base level change that we expect to see on a chemical level. And that's kind of the cool thing about evolution, is that yes, there's the big fancy evolution, look at all the diversity in animals, but at the end of the day, like everything in this class, it's because of chemistry, and that chemistry happens in a somewhat predictable way. And so we can look at the mathematics behind, like, what's just the expected rate of mutation of different genes over time. And ultimately that becomes the basis of sort of the next chapter, which is all about phylogenetics and things, where we're able to then look at how many differences do we have between these organisms, what's the rate of mutation that we expect to occur, and then we can start working backwards to figure out when about was the common ancestor between these two divergent pathways that lead to humans and blobfish, based on the differences that they have in their DNA because of well-studied rates on the chemistry behind the creation of those mutations. The lysozyme is kind of a cool example your book goes into. The lysozyme is something that digests bacterial walls, and some mammals are called foregut fermenters. And basically what happens is they rupture the bacteria. They use lysozyme within their stomachs. So they have bacteria in their stomachs. They eat grass, the animal, like a cow, eats grass. The bacteria helps to break down the grass because we can't. We don't have like the right enzymes and sort of chemistry in order to break down the tough cellulose. And then they use this lysozyme to break open the bacteria and release the nutrients that have been broken down by the bacteria. And so that's the whole thing of like cows, they chew grass and then they like throw it back up into their mouth and they chew on it for a while and they swallow it back down. That's this whole process, which is kind of cool. And it's a neat example of evolution that would lead to that. And specifically, it's pointing out the fact that this has actually happened 2 times in very similar ways in terms of the mutations. Kind of like when we looked at the stickleback and how that has happened countless times in different places in the world, but in exactly the same way. And so there's some reliability, again, from the basis of the fact that this is all chemistry. There's reliability in terms of you just need the right conditions to find a way to push a population in the right sort of way. And these mutations will happen. It's just a matter of, do they benefit the organism in the right way at the right time in order to sort of take hold. And so these different Langer monkeys and cows actually share a significant commonality in terms of their digestion of their food, not on the basis of shared ancestry. It's not like, these monkeys and cows had a shared ancestor who started it and then it carried forward, but actually it appeared in both of these lines independent of one another, but in the same mechanism. So it's kind of cool. And this is like the differences in the amino acids, I think, or nucleotide substitutions in a particular gene in all these different organisms. But here you see the Langer and the cattle have 5 shared nucleotide substitutions that are key to basically making the bacteria able to survive in the pepsin environment of the stomach, so that the enzyme, that lysozyme, is able to survive the conditions of the stomach. It's kind of cool. Genome sizes vary significantly, and that also is a factor in the mathematics behind looking at the rate of mutation between different organisms. The book goes into this. You won't be asked about genome sizes, but just understand, yes, there's significant differences between the size of the genome of the E. coli versus a human versus any other organism. And that's not just number of chromosomes, but actually number of nucleotides, because chromosomes can be big or small. I included this because I think this is interesting. Our book talks about much of the non-coding DNA does not appear to have a function. You got to really read into that sentence, does not appear to have a function, but more and more every day, like this is a huge topic in biology right now and in genetics, is the understanding that the junk DNA that we often long ago sort of referenced as just like, it's garbage, it doesn't do anything, serves a whole variety of purposes that are really just not yet well understood. So that's also a major factor in evolution. And I think we're sort of repeating ourselves here. So the last section of this was talking about the different types of reproduction and why they exist. Sexual reproduction brings about a whole bunch of advantages in terms of just new opportunities for fundamentally different allele combinations, as you are bringing 2 individuals' genes together to make a wholly unique new organism. There's some disadvantages listed that come down to sort of the chemical and biological sort of consequences of that type of reproduction. But ultimately, the book goes through the whole case for like, well, if there's disadvantages and it can be tricky and causes problems, like why does it exist? It exists because of this opportunity for drastic change, which allows then for selection to have something to take hold of and favor over time, which ultimately is a win for the population, even though it may be for some individuals a negative. If you're born with the wrong set of genes at the wrong time in history, my terrible eyesight or whatever, that could be a really bad thing. That does not help your survival, and that's rough for the individual. I guess it's good for the larger population because, Yes, that means somebody with better eyesight, didn't get eaten by a saber-toothed tiger or whatever, a million years ago. But anyway, the book goes into more detail about that and sort of the pros and cons between asexual and sexual selection. But don't get tricked. While we are more complex and I think interesting than bacteria has made it just as long as we have in terms of evolutionary history. So different organisms have different requirements, and so we don't want to get in the trap of thinking of something as like more evolved or less evolved. Humans are more complex. We have TV and internet and fun things like that. But at the end of the day, if you're a bacteria, you probably don't care about a lot or anything, and you just sort of reproduce asexually and do the whole lateral transfer of genes thing, which I think is the next slide. And that's just the way in which exchange of DNA can happen between organisms. But basically, they survive just like we do. And so evolution, has steered different organisms to a successful path. And anything that's alive today, you can point to and say, all right, evolution did okay for you because you're still here existing in the world. Cool. Last little bit was just about sort of the practice of this. We have breeding programs and things, understanding like pesticides and how do we control for different mosquitoes and bugs that cause problems to us in a variety of ways. And the last little bit here, I think we're over. We are well versed in the understanding of the genetic analysis of things like viruses and strains and things like that. So a lot of practical applications to evolutionary theory. If you have questions, please let me know.